This figure you can get is extracted from Campbell 12 edition, page 393 on transcriptions. So this figure shows all the 3 stages of transcription process, which are initiation, the second stage is elongation, the third stage is termination. Okay, start with the initiation. So they're going to be a promoter first to start off with the transcription. That's a start point, which consists of a short sequence of nucleotides with the bases T and A. So we're going to take the 3' to 5' as the template. So 3' to 5' is known as antisense or negative strands. And because the synthesis for transcription always occurs from 5' to 3'. So when the transcription factors, the transcription complex binds to the promoter, the starting point, so that will enable the RNA polymerase to bind and to start or initiate the transcription process and start synthesizing new strand. This is initiation. So RNA polymerase will unwind this double helix DNA and form a transcription bubbles, right? That's the initiation process. And then the RNA transcript starts to be synthesized. There's no primer needed. And then this is replication bubbles, and RNA polymerase starts to add the RNA nucleotides, so ribonucleotides, to the 3' end. And that will cause the RNA transcript to elongate. So elongation occurs here. So it will elongate. And then after elongate, if it's long enough, then it will be separated from the DNA, okay, template. So it will be separated that and reach the stop signal. So this is termination. So this is the completed RNA transcript with coding and non-coding parts. The coding segments of the RNA transcript is known as exon, and the non-coding part is intron. So these RNA transcripts need to undergo another stage, which is known as post-transcriptional modification, where the intron is removed or eliminated and the exon is joined, or the RNA splicing occurs during the post-transcriptional modification. Apart from that, at this head part, 5', we'll be adding the GTP, and at the 3', the adenine will be added. So that will be in the post-transcriptional modification. So straight away, I'm going to go to post-transcriptional modification. So at the 5' caps, as I mentioned earlier, see, this one is 5' and 3' here at the tail. This is like head and tail. So GTP will be added. So 5' linkage is formed with GTP. So forming 5' cap. Dia akan ada cap kat situ. Okay, that is one of the stage of the post-transcriptional modification. Apart from that, apart from forming the 5' cap for post-transcriptional process where a modified guanine nucleotide added to the 5' end, the 50 to 250 adenine nucleotides added to the 3' end. When 50 to 250 adenine, or A, is added at the tail end here, this is known as poly-A tail. Poly means many. So you have the cap and you have the poly. And in the middle here, so we're going to be segments, coding and non-coding. So we move on. That will undergo the splicing. So exons is the coding segments. Intron is non-coding segments. So initial transcription produces primary RNA transcript, the initial one. It has coding and non-coding. And then we'll need a proteins known as splicing proteins to cut the intron and joins together the exon, forming shorter, mature mRNA transcript. This process is known as RNA splicing. Okay, so RNA splicing is joining the exons of the coding part with the help of splicer, which is the proteins, and cuts or removes the introns away. So this is the RNA splicing. Okay, RNA splicing, whereby in this RNA transcript, it consists of exon. The red one here represents the exon. The yellow one represents the intron. The exon is the coding part. Yellow is the non-coding part. So the non-coding part, which is the intron, is cut and removed by a protein known as splicing, and the exon then joined together. So it formed like this. Okay. Apart from that, of course, there's going to be a adding of the GTP at the 5' and adding about 250 adenine or A at the tail. So you form the 5' cap and 3' poly A tail apart from the RNA splicing in the middle. So you produce the mature mRNA transcript here.